Hi everybody, my name is Christoph Sperber. I'm from the University of Tübingen at the section of neuropsychology, and there I work as a postdoc with Professor Hans Otto Karnert. I finished my PhD 2018 and 19 on the topic of lesion-brain inference and multivariate lesion-brain inference, and that's going to be the topic of my talk today. First of all, thanks a lot to Laura Hokkanen and the organizers of this alternative event to the FESN Summer School. I'm very happy to be part of this, and I'm also happy to be able to present my work, my topics to you. So, these are the topics of my talk. I will start with a short introduction into lesion behavior inference and univariate statistics. So this is the way lesion behavior mapping has been done roughly the last 20 years. And this approach has some limitations. This is why some researchers started using multivariate lesion behavior mapping instead. And I will show you the basic ideas behind multivariate lesion behavior mapping, and also my two favorite methods in this field. The last part will be a critical evaluation of multivariate mapping. Does multivariate lesion behavior mapping really work as intended? Does it hold all its promises? Well, short spoiler, no, it does not. There are still some problems with it, just like with the univariate approach. In lesion behavior mapping, we want to understand the functional anatomy of the brain, and we do so by looking at patients with neurological disorders, for example, with stroke. And these patients have certain disorders, for example, language disorders, and we want to map these functions, like language functions, onto the brain. There are quite many methods available to map functions onto the brain. Besides lesion behavior mapping, that is always a little bit complex because you need access to the patients, there are methods just like fMRI, EEG, MEG, or neurostimulation. However, lesion behavior mapping is still a hot topic today and is widely used. Have a look at this review by Ron and Karnert from the year 2004, where he made quite good points why it is still important. Short note, all the methods that I discuss here are based on normalized lesion information. So we get highly individual brain scans that depict brain damage, and we normalize them onto a template so that in the end they look quite the same in regards to shape and positioning of voxels. It means a voxel at a certain coordinate in all of these brains should represent the same brain area. And we also have the lesion area somehow delineated. For example, with manual, with semi-automatic or automatic methods, we identify the brain area that is damaged, and this is the lesion map. After normalization, it's the normalized lesion map, and this is what we require for all these methods here. For a long time, the most popular approach to lesion behavior mapping was based on mass univariate statistics and on statistical parametric mapping that investigates for each voxel in the brain image individually the relation between brain damage and behavior. So I just called it mass univariate statistics. This talk is about multivariate statistics, so I guess I have to explain what is univariate. Univariate statistics usually just look at a relation between one variable. This could be drug intake, I take a drug or take a placebo instead, and one outcome variable. For example, blood pressure. And univariate statistics always look only at two variables, but it's not called bivariate, it's just like we call it univariate because it's one dependent variable. How does this look like in lesion behavior mapping? There we have the independent variable, that's the brain damage, but we only look at brain damage in one single spot in the brain, a so-called volumetric pixel or voxel. And we look at the relation between the damage status of this voxel and a certain neurological disorder. The statistical tests that can be performed here, and also in the example before, are for example t-tests or Brunner-Munsell tests. Now, how does this exactly look like in a mass univariate test? We just investigate one voxel here in this one test, and we have a large amount of patients tested. Now, some of these patients have a lesion in this voxel. like those marked with red, and others do not have a voxel in this lesion. These are the ones in purple. And for each patient we have a continuous outcome variable, for example the language score. And with these variables we have a grouping variable and a continuous target variable, so we could for example apply a t-test. Such a statistical test is done for one voxel in the brain, and the same for another voxel, and another one, and another one, and many more. So you see, this is why it is called a mass univariate approach. We do a mass of univariate tests up to maybe even 500,000. In the end, we have a statistical map. For each point in the brain we have a statistical value that indicates the strength of the relation between damage status and behavior. By the way, this map should be corrected for multiple comparisons. We do statistical tests over and over again, so we have an accumulation of the alpha error and have to control it. The final idea in this analysis is that we find some voxels where the presence of damage is associated with higher deficit, and we assume that this voxel is part of a cognitive module, or the neural correlate of the function that we investigated. Let's get to the one major limitation of the method. Imagine you do one test and you do many more tests. The second one here. Both tests are conducted independently. This is the one important thing that we have to think about now. So what does it mean that both tests are conducted independently? Well, let's explain from the other side. Imagine you test a brain area like the STG, and it has 1000 voxels. You test 999 and we find each and every voxel to be significant. Now there's only one voxel left, voxel number 1000. Would you bet €50 that this voxel is also significant or not significant? I bet you would, because you do not think of all these voxels to be independent. The tests, however, they just behave like nothing happened before and do the same maths like in the 909 tests before. This assumption of independence between voxels, however, might not really fit the brain and its organization. First, the brain is thought to be organized in largely cognitive modules or brain networks, where many brain areas and many voxels somehow are connected in producing a certain function. The other thing is that brain damage does not happen to voxels independently. Here you see an example of the posterior cerebral artery, and this artery has a typical anatomy, and where a stroke exactly happens is always at quite typical places. So in the end, the anatomy of stroke in general looks quite systematic, and there are areas that are more often damaged, and many areas are systematically damaged together or not damaged together. This means independent testing in the mass univariate lesion mapping approach contrasts to the brain anatomy and the brain organization in two ways. This is the functional dependence between voxels and a lesion anatomical dependence between voxels. Does this actually affect mass univariate analysis? Do we get somehow wrong results? Well, yes, it looks like this is a problem. First, the functional dependence of voxels. This has been discussed in the context of the so-called partial injury problem. Have a look at Roden et al. 2009, or in my paper where in 2019 in HBM, where I tried to provide a simple explanation of this issue. And this issue states that if we have larger functional areas or brain networks that are often only partially injured, so where lesions only affect parts of it, then we have a lower statistical power and we might miss some areas. So here we have false negatives. The other issue is the lesion anatomical dependence. Madal 2014 or Inoue 2014 likes to put a finger at this issue, and it states that due to the systematic anatomy of lesions, we might find false positives, often just next to the true neural correlates and somehow skewed towards the centers of the cerebral arteries, but maybe even in other areas, like it has been shown by Madal. So this is really bad. But is there a solution? Well, obviously now I got to do multivariate statistics. It has been claimed that multivariate statistics, that means not conducting 2 independent tests, but only one test, or to only compute one single model for the information of several voxels, should overcome this issue. First of all, a very short definition on what is multivariate statistics. In Wikipedia, it says it is a subdivision of statistics encompassing the simultaneous observation and analysis of more than one outcome variable. So going back to our example, it would look like this. 2 outcome variables, blood pressure and blood sugar. But this is not the way we use multivariate in lesion mapping. It rather looks like this, with a larger amount of independent variables. More exactly, in lesion mapping, we have the information from many voxels or areas in the brain, maybe even up to many thousand, and as an outcome variable, we have a measure of a behavior, or a deficit. We have to go a little bit deeper. So, on the one hand, we have input data, these are binary lesion maps, and we have output or target data, this is a behavioral score. The numbers indicated in red here are the measurements obtained in only one subject. So for each voxel number and one outcome score. At this point, it's not difficult to just use a machine learning algorithm to train a model to predict output from input data. This could be a super vector regression. So we have continuous outcome scores here, so it has to be a regression here. And it could also be a Gaussian process regression or any other regression method. The problematic thing here is just training a model is not the whole thing. It could also be a bad model. If you want to find out if a model is very good, you have to validate this model. And this works in the following way. First, you train the model on your training data. For example, the data you see here, this could be training data. And there you obtain a model. And this model is applied on the new data. Here, these are the validation data, 3 subjects. And for these 3 subjects, this model makes some prediction. This output could be 3, 2, 4. And secretly, we already measured the true score in these subjects. And we know it is 3, 3, 2. So, if this model is really good, the predictions of this model in these previously unknown subjects and the actual data should highly correspond. So just in short, again, we have training data where we have input and target data. We train a machine learning model with these data. Then we have validation input data. We apply the model that we got from the first step and predict the outcome data. And we have the true target data, and we see if the predicted and true target data here in this validation sample correspond. This strategy has been used, for example, by Rondina et al. 2016. They looked at different kinds of representing lesion information, so also binary lesion maps, and wanted to look if a certain kind of representation is best suited to predict post-drug motor outcome. They used Gaussian process regression and super vector regression and found some models that provided quite good predictions. Another study is the one by Siegel et al. They looked at patients where they had both lesion data and functional connectivity data, and they found out that depending on what deficit you look at, the models to predict are better either with lesion data or with functional connectivity data. It differs between different deficits, and thus in some situations lesion data or functional connectivity data might be differently suited to make predictions. Well, this is super interesting. I really like these studies, but unfortunately this is not lesion behavior mapping. Remember, lesion behavior mapping was about mapping the functional anatomy of the brain. There are several studies in the field that applied methods that map functions onto the brain using multivariate algorithms. But they have limitations. For example, some of them have been limited to only a small amount of anatomical features, mostly due to computational limitations. Others were restricted to an a priori population of brain areas. This is not necessarily bad, but if your brain population is bad, results might also be bad, so you introduce a bottleneck here. One study lacked the actual inference, and most studies were not validated. This is more an optional point, but quite important to me because you have means to validate a method to see does it really do what it's supposed to do, and many studies in the field just introduced the method, applied it to some function, interpreted the mapping, and at this point the whole method was established. Still, it might not be that good as expected. In the end, two methods are left that I really like, and this is the SVR LSM by Zhang et al. And the SCCAN in lesion mapping by Pristina et al. Let's get to multivariate lesion symptom mapping using stupid vector regression by Zhang et al. 2014, published in Human Brain Mapping. We can start right away with the same visualization of the data with input data, output data, and a machine learning model that is here a stupid vector regression that has been trained to find a relation between both. As you might remember, just training a model is no statistical inference, nor it is any kind of lesion mapping. To get there we require an additional step, and this is the computation of feature weights. If you want to know what these feature weights mean, think of the beta parameters in a regression. The feature weights in this situation are often called pseudo-betas, and they also indicate for each input variable, so here it is for each voxel, how much this variable contributed to the final model. These features can be computed for the actual data. Still, there is no statistical significance yet involved. To get there, we require one more step, and this is permutation testing. We can permute the original behavioral data, and this results in some kind of nonsense data without true signal, and also compute feature weights. We do this once, we do this twice, and many more times. This follows the standard scheme of permutation testing, and in the end we in fact have a probability for each voxel that indicates if it significantly contributed to the overall model. There are a few caveats involved, so be careful. First, the contribution of each voxel is tested individually, resulting in many tests, so again we have the multiple comparison problem. Then there's an issue with kernels. Usually you can only compute feature weights in linear kernels, but it's anyway done for RBF kernels. Zhang et al. Showed that this is asymptotically correct, but they also assumed that you use the lesion size control they used, otherwise it might not be correct. And last but not least, hyperparameter optimization is quite difficult here, how to evaluate hyperparameters, and there is no out-of-the-box software that does this straightforward. There are some scripts available by Dan Merman that do the hyperparameter optimization, but still to evaluate it is quite tricky. There are some toolboxes available that provide you with everything you need to perform such an analysis. The first thing is the collection of scripts by Yongchen Zhang, the original paper. It's available on GitHub, and another one that I really like is the SVR LSM GUI by DeMark and Turkeltaub. So this provides more or less the same thing, but also with a user-friendly graphical user interface. So this is what you get when you decide to download the scripts by Yongchen Zhang. This folder contains a manual, well, read it, the main function. This is what you have to run in MATLAB to make the magic happen. Then there is libsvm. This is just a tool that allows you to perform SVR or SVM in MATLAB. Importantly, you have to make sure that it's installed at the beginning. You might need a decompiler to do so. And there you have the functions. Here are the functions that are executed after you run the main file. The main file knows where to find them and will use them accordingly. And there are some important functions that you might want to have a look at. It is first to read lesion images and read behavior score. And then quite importantly is the get beta map and the run SVR LSM beta map. This is what the main script SVR LSM toolbox looks like. There are a lot of parameters that you can change, like paths to the data, to libsvm and the functions, then a path to a score file, and several parameters. Make sure that you read the paper and understand what these parameters do, so you can change them accordingly. And then the last thing that is required is just hitting run. This script will use the other subscripts, for example, run SVR less than beta map, and this is an important point here in CMD. There the parameters for the SVM are defined, so if you ever want to change something like the kernel, do it here. And in get beta map, this is applied, and this is the line where the SVM happens. This is the line where the parameters are computed. So if you ever want to change something, or if you want to understand the script, start looking there at the basic functions. If you are not in the mood to jump into such a complex collection of MATLAB scripts, then you might instead go for the toolbox by DeMarco and Turkeltaub. They wrote a nice paper on the method and on the toolbox. They show how it works and how it compares to the original scripts by Zhang et al. And the toolbox is really easy to use. Look online, they have some manuals. They also nicely explain the prerequisites that you have to install to use it. And then it's just straightforward, a little bit the same like the scripts that I've shown you, but with a graphical user interface. It makes it more intuitive. And this is the second method to multivariate lesion behavior mapping by Pristina et al. 2018, based on sparse canonical correlation analysis for neuroimaging data. This one works quite different than the super-vector regression-based lesion symptom mapping. Again, we have the same input and output data, but we do not include all input data in computing a model. The sparse canonical correlations instead look for the voxels that have the highest correlation with the output data. And by doing so, the analysis finds the voxels that provide the best model. So consecutively, there's a model built up with voxels that are good at explaining the output, and in the end, we only have one single model. And we can test the model by checking if the prediction of this model is good. I advise you to have a look at the paper by Dorian Pristina. It's a great paper and an even greater evaluation of the method. And they also published a toolbox. It's the Lesion Map toolbox, and this runs on R programming language. A little bit tricky is it only works on Linux, but on Windows 10 there is the Linux subsystem per default installed, so this might help you. If you get it installed, on this page you find some instructions how to do it. The dependencies can be quickly installed with this function. Might give some problems, but if this works, then it's straightforward, and then you can just easily use the lesion map functions to do a lesion mapping analysis. There were also some tutorials here, some manuals. Have a look at them. They will probably help you to get lesion map running and also to use it. Great. So it looks like we have these two great methods to perform multivariate analysis of lesion behavior data ourselves. Does this now finally mean that we can overcome this problem in univariate analysis of independent testing? So remember, we had the functional dependence between voxels that was not considered by univariate analysis, and the lesion anatomical dependence between voxels, also not considered by univariate analysis. Does multivariate analysis consider these two things? Let's see. As you might remember, there are some methods that allow us to validate if a lesion symptom mapping method is good, and also especially if the lesion symptom mapping method can overcome the problems with functional or lesion anatomical dependence between voxels. This has been done, for example, by Ma et al. 2014, Zhang et al. 2014, or Pristina et al. 2018. And it looks like the functional dependence between voxels can be accounted for by multivariate models. Let's look, for example, at Zhang et al. 2014. They investigated neurocorrelates consisting of 3 areas, so it's a little bit like a brain network here. And the multivariate approach was better at identifying the neurocorrelates in each of these parts of the network. Univariate analysis might have had some false negatives. The same in Pristina et al. Here, the SCCAN approach was also better in finding multiple brain areas. Anyway, of course the multivariate approach is not perfect. It can still fail to find some areas, but in most situations it is better than the univariate approach. So all in all, it looks like, yes, multivariate methods can overcome the problem of functional dependence. On the other hand, I did a study where I decided to not include any functional dependence. I did so by going for a simulation approach where I decided to just use one single voxel as the neural correlate of the function. So there were not two or more voxels working together to produce a cognitive function, always just one. And I visualized the results with these little vectors. You can see them here. They always point from the actual true voxel to the center of the statistical map that resulted. This is the same way of analyzing the data like MAD out 2014 did it. Ideally, the vector should be short. The results should be centered on the voxel itself. Madel found that it's not the case, and you can see this in A. Here, the voxels in the brain most often show towards the center of the cerebral arteries, so results originating from this point are misplaced in this direction. Now, this is the univariate analysis. In C, on the right, you see the results for the supervector regression-based lesion symptom mapping, and the direction of the bias was more or less the same. There was no statistical difference, and the amount of bias was also not statistically different. This means supervector regression-based lesion symptom mapping had the same bias as the univariate approach. This is a point that I recently made in an empirical and theoretical paper where I claimed that no current method for lesion behavior inference, be it univariate or multivariate, is able to overcome the issue with lesion anatomical dependence. All results will be biased by this problem. So, if you want a full-blown explanation why all these methods are incapable to overcome this issue, you might read the paper. But let me get to the main point. Let me explain it to you with a very simple experiment. And this simple experiment involves this object. Obviously, yeah, my favorite color. Spending many years in science, well, sometimes you forget about how things work outside in the real world. You just spend your whole life sitting in front of a computer in a lab, and, well, there were maybe more important things, like this object. I think it has been called a candle, and my parents always warned me of using it. There was something dangerous, painful. I can't exactly remember it. I just remember how it's being used, somehow in this way. Yes, and we see there is a shining bright light. Beautiful, isn't it? But I remember it's been painful. My parents always warned me, so I guess this light might be painful. Light might induce pain, right? So let's do a short experiment if a candle's light induces pain. Try 1. Try 4. Try 17. Try 25. Try 47. Try 50. Okay, welcome back. So, the first question you might have is, what does it have to do with lesion mapping? What is happening? Wrong question. First of all, we get to the data analysis. Here you see the results of the study. I did 50 trials, and in 25 there was a light, that's a 1, and in 25 there was no light, that's a 0. And I assessed the pain level with a pain assessment scale ranging from 1, no pain, to 9, maximum pain. If we look at the average values, we see that in a no light condition the average pain level was 1.4. In the light condition the average pain level was 8.6. I've thrown all this data into a t-test, and this became highly significant, indicating that there is a relation between light and pain. In a second analysis, I realized, well, just doing a univariate test is not state of the art anymore. I'll have to do some fancy thing with machine learning. So I measured light continuously and binarized pain level. The blue balls are trials where there was no considerable pain, and the red balls indicate trials where there was considerable pain. I trained an SVM classifier that found a hyperplane in the space, and this provided a perfect classifier with 100% prediction performance in a leave one out cross-validation. That means the light level can be used to predict pain. So it looks like we just have proven that light induces pain if you touch something that emits light. Probably it's quite the same like in Star Wars with the lightsabers. You hit somebody with a lightsaber, probably it's awfully painful. And now back to serious. Why have I shown you this stupid experiment? Why did we analyze it? What do we learn from it? The thing is, you intuitively understand that something's just wrong. We messed up with the experiment at some point. You know, it's not light that induces pain, it is heat. And the other important thing is, we did the experiment quite the same way we do experiments in real science. There was nothing that was completely odd. Yeah, there were some things, but not the central things. So, how do we usually do studies? We also have a hypothesis. We measure some data, we analyze them with univariate methods or with some multivariate methods with machine learning or whatever. And in the end, we interpret this data, often in a way of looking at causality. And that's the very problem, the causality thing. Let me explain you. The straightforward interpretation of our data would be that light induces pain. And we can visualize this by this very simple causal graph where the arrow indicates a causal relation. And obviously, it's not like this. Often, in papers, we try to have stories that are straightforward, that are easy, where one variable induces the other one, but in reality, things often look different. Here, it probably looked like this. So we had fire on a candle. This induced light, and on the other hand, it induced heat. And this heat induced pain. And now, the big question, what does it have to do with lesion behavior mapping? In lesion behavior mapping, we also have a problem of causality. Think of a simple situation like this. We have two brain areas, A and B, and they are both supplied by the same minor cerebral artery. So, most often, if there's a stroke affecting one area, there's also an affection of the second area. Areas A and B are often damaged together. Now imagine only A is the neurocorrelate of a behavior. If we now do lesion behavior mapping, you will probably also find that area B is often associated with the symptom, like light was associated with pain. So, what I wanted you to understand from this little experiment is that whatever statistical methods we use to understand data, be it something univariate, be it multivariate, be it some machine learning algorithm, whatever we do, these methods are not able to truly understand causality. If we understand causality with such methods, it's only because we chose a certain design. If we do an experimental study, well, then we can understand causality, or we already have some hypothesis, like in the candle example, we somehow understood no, it is not the light, and we did so because we already had some hypothesis what our body can perceive. And now going back to our lesion mapping example, note that in the candle example we had perfectly deterministic relations. In lesion mapping there are no perfectly deterministic relations, but this is not necessary. Causality is still a problem, even if relations are not perfect. There's also another issue with causality. Some scientists claimed that in order to understand the brain and its functional anatomy, we need to acquire highly predictive multivariate models. Now, my point is, having a predictive model doesn't necessarily mean that we have understood the brain, and we can see this by this very simple example. Imagine there is a cognitive model, this is the yellow area, and if it is damaged in large parts, then a deficit arises. This area is supplied by a major artery with two subarterial branches. Now there can be stroke in one artery. This leads to damage only in parts of the area and the deficit does not arise. So the neurocorrelates are damaged, but there is still no deficit. The same with a stroke in the other sub-branch. However, if there is damage in the supplying artery, then the whole area is affected and a deficit arises. And here we see damage just inside the neurocorrelate of the function is not necessarily highly predictive of a deficit. However, information taken from outside the neurocorrelate can be highly predictive. And if we have something that is good at predicting a symptom, it doesn't mean that we exactly understood how the brain works. This is a question that I looked into recently. There's a preprint available on bioRxiv, and they are just asked the question, where in the brain should I look for information when I want to predict motor outcome? The intuitive answer is, look in the primary motor system. Unfortunately, it's not that easy. It's more complex. There are more areas in the brain that are able to predict motor outcome. Okay, so as a conclusion, multivariate lesion behavior mapping is a good thing. You can overcome the problem of functional dependence, and you are more likely to find the full neurocorrelates of complex cognitive systems or brain networks. On the other hand, it's not perfect. It still suffers from biases introduced by the lesion anatomy. And if you look into the field, there were a bunch of scientists that said that we should use multivariate algorithms just to overcome this problem. And I would say, no, they are wrong. And I hope now you got some intuitive understanding why multivariate algorithms are not perfect, why no algorithm so far is not perfect. Maybe one day we might have some kind of causal inference overcoming this issue. I'm not sure. I don't know how it could work. Anyway, I like the methods. They are quite good, although they have some problems, but the same is with fMRI, with EEG, it's not perfect. And all these methods nicely complement each other. So, that's it from my side. Thanks for your attention, and I'm looking forward to your questions.